Next, we're going to talk about the cross-bridge cycle. We'll look at the role calcium plays in activation of that process of the cross-bridge cycle, talk about the activation of the myosin head, and then I'm going to walk you through the steps. To do that, I'm actually going to go over to this page here where I've done some drawings, simplified. This is similar to what I would do on the board if I were teaching this in class. So what I've got here is a simplified drawing. So the diagram that you can see here, if you focus on these purple circles here, these purple circles are my actin. Okay, that's what these circles here are. This is actin. And you notice I've only drawn it in a straight line. I'm not doing the helix. Actin is usually in a helix structure. But for my purposes, we'll just simplify that and keep it kind of just a straight line. So each of these balls represent a globular actin, and then together they make up the actin filament. The orange line is my tropomyosin. It is a rope-like structure, and what it's doing is it's wrapping around actin, and it is shielding or blocking the binding site on actin for the myosin head. The yellow circles are my troponin, and it sits right there on top of the tropomyosin, and I like to think of troponin as the governor. It governs the tropomyosin and dictates whether tropomyosin is capable of binding actin so that it blocks the binding site or not. Then if we look down at the structures I've drawn in blue, what we have is myosin. And again, this is simplified in that for obvious reasons, I'm not drawing all the hundreds of heads. Instead, I'm showing just two myosin in two different positions. When we're at rest, so this is the muscle at rest, the myosin, most of the myosin is not attached to actin. You do have a few myosin heads that will manage to attach. There's competitive inhibition here, and so sometimes myosin actually wins the spot, but not a lot of them are bound. Most of them are in unbound state, and they can either be in the forward position, as shown here, so this structure right here in forward position, and you can see it's at an angle to the actual, well, to the horizontal line there. And then we have the cocked position. Now, the cocked position references, in general, its purpose is to reference this position in a state of readiness to initiate what is known as the power stroke. And that cocked position, when we're not bound, is pretty much useless in that it's not if it's not attached to actin, we can't generate any kind of force. Notice that we have a DP, an inorganic phosphate, bound to myosin when it's in the cocked position. This process of moving back and forth between forward position and cocked position is what we sometimes refer to as myosin activation. The steps for myosin activation, if we look at this, let me zoom out a little bit so we can see all the steps, is what really triggers this process. It's when the myosin is in the forward position, ATP is going to bind that myosin head. That ATP is very quickly hydrolyzed. So once that hydrolyzation occurs, so if we look at these steps, myosin in the forward position, ATP binds myosin head, ATP is hydrolyzed to ADP and inorganic phosphate, and it's this hydrolysis that releases the energy necessary to move the myosin head into the cocked position. And so we get this movement into our cocked position like so, but then the ADP and the inorganic phosphate, they're going to leave. And so as these molecules detach from myosin, we move back into that forward position. So the process of myosin activation is about getting it back into that cocked position. We're going to look at that process and how it affects muscle contraction. Now we get to the point where calcium is needed. So when we're ready to initiate a muscle contraction, we're going to get an action potential in the muscle fiber. That action potential leads to the release of calcium from the sarcoplasmic reticulum, and that calcium, what calcium's going to do here is it's going to bind to troponin. And we end up with a troponin-calcium complex. So troponin, sometimes written like this. And it's this complex with the calcium bind. If I zoom in a little bit here, you can see I've kind of written in calcium there as it binds to that troponin complex. Now, as often happens when a ligand, in this case the ligand is calcium, right? When a ligand binds to a larger molecule, in this case troponin, which is a protein, it causes a shape change. And so calcium induces this shape change, and troponin changes shape. Troponin is attached to tropomyosin, and so we end up getting kind of a transfer of that shape change And as troponin changes shape, it pulls or tugs on the tropomyosin and tugs it out of the way. And this exposes the binding sites on actin so that the myosin can now attach. So myosin then will bind to that actin, and this structure right here between the myosin and the actin that I'm highlighting here in yellow, actually let me switch to pink, something that doesn't contrast with the troponin. This is what we call our cross bridge. This is my cross bridge right here. And the more cross bridges we get, the stronger the force that we can generate through this molecular process. And this is also the source of the name. This is why we call it the cross bridge cycle. Now, once myosin binds to actin, we're ready to move on to the next step. And what happens here is, if we look at this, so myosin binds actin, the next thing that happens is that we end up with myosin losing its inorganic phosphate. That phosphate leaves. Now, the process of the phosphate leaving has two very important functions. One, it strengthens the attachment between myosin and actin, so that attachment becomes quite strong. But two, it causes myosin to change shape, and myosin moves into that forward position, but this time it stays attached to actin. And so while attached to actin, what it's going to do is it's going to drag that actin forward. And so if we were to look at this actin filament, so we're going to kind of scribble in a little bit of purple color here to kind of track this particular molecule of actin. So when we actually move forward, that molecule, this one here, is going to be dragged with that myosin head several steps, so that when we've finished that power stroke, that's what we call it, the movement into the forward position, then we're essentially transferring the purple molecule forward, and the entire filament of actin slides towards that M line. Now, notice that actin stays attached. We still have this attachment point right here. Let me switch to pink again, sorry. This attachment right here. We still have that cross bridge forward, and myosin no longer has ADP bound. At some point during the power stroke, ADP detaches. There's conflicting information, and I really honestly haven't dug into it enough to be able to answer the question. I don't know whether ADP leaves during the power stroke or immediately after the power stroke. So we'll just say at some point during that power stroke or at the end of the power stroke, ADP is going to detach. Now, when the myosin head is stuck in this forward position, if it's attached to if it's attached to actin in this forward position, strongly attached, we have a different name for it. We call it rigor state. And hopefully the term rigor reminds you of a closely related term called rigor mortis. Mortis? Probably spelled wrong. That's all right, we'll work with it. Rigor mortis. And rigor mortis occurs after death, and what happens with the muscle fibers is, as ATP is depleted, we end up getting those fibers stuck in that rigor state, and so we get the stiffness of the muscles curling, and so forth. So the name rigor mortis is related to rigor state. Now, so how do we break rigor state? This is quite a strong attachment, and that myosin head is going to remain attached to that actin fiber in rigor state. Well, to break rigor state, what we have to do is attach ATP. So as ATP is attached to the myosin head, that action of ATP binding myosin changes the shape of myosin once again, and myosin now detaches. So to break rigor state, we need ATP, which is why during death, as we get this spontaneous release of neurotransmitter and spontaneous action potentials being generated in the dying neurons and the dying muscle cells, we go through a cross-bridge cycle, making those muscles contract. The limbs curl up, the face goes into a Richter state, and that is occurring because of the spontaneous contraction of the skeletal muscles. But then eventually it's going to get stuck in that rigor state when we deplete the ATP. Now, once ATP attaches to myosin and myosin detaches from the actin, we're ready to repeat the cycle. And that cycle will repeat as long as calcium is present in the sarcoplasm. Remember, sarcoplasm refers to cytoplasm of skeletal muscle. So if calcium is present, it's going to repeat this cycle. So we'll start right back over again with the initiation of the cross-bridge cycle with myosin binding actin. So ATP is hydrolyzed into ADP, myosin binds actin. We have phosphate leave, we execute the power stroke, we're back in rigor state, ATP binds, and then ATP is hydrolyzed, and we're right back into this original state here. And so that process continues onward until calcium is removed. Now, once calcium is removed from the cytoplasm, we're going to go back into its resting state. Tropomyosin is going to glide back over the binding sites, and we go back to that resting process. And that's, in essence, the cross-bridge cycle. Now let's take a look at it in the slides so you can kind of relate what I just told you to the slides that I show you. The first thing that you will notice as we look at the slides in the PowerPoint is that the orientation is different. So let me make sure you understand how things are oriented. If we consider a simplified sketch of a sarcomer, for example, we know that a sarcomer goes from Z-line to Z-line, that actin radiates from those Z-line in toward the center. So here's my actin fibers. And that is going to be true from both of my Z-lines, that we're going to have this actin fiber radiating in toward the center line. Now, the amount of overlap will change, of course, so let's actually reduce this overlap just to make it seem that we can actually contract this muscle to some degree. And then if we look at the myosin fibers, we will see that the M line found in the middle here, and the myosin fibers radiate out from that M line. And then we can actually draw the myosin heads onto these fibers. So if I were to draw a head here, it would attach to actin like so, if I'm going to have the cross-bridge cycle. But over here, we're going to have attachment to actin like so. And then down at the bottom, it's essentially the opposite direction. And the result is when these myosin fibers contract, so as we execute the power stroke on the right side, that's going to tug the actin inward toward the midline. And remember that the midline is this line right here. This is the midline, M line. But then on the right side, it's going to go in the opposite direction. Now, by habit, just as I've drawn this so many times for different classes, I have a tendency to diagram essentially one side of this, whereas the figures that I sometimes use often show the other side of it. So if we look over here to the figure on the left down at the bottom here, what we're actually going to see is this is the muscle in its relaxed state. Let me illustrate that here. Relaxed state, troponin does not have any calcium attached to it, and tropomyosin is blocking the binding site. So let me highlight those binding sites for you so you can see them clearly. The binding site right here on the actin filament, this is where myosin would attach if it were capable of accessing that point, but that binding site is hidden by the tropomyosin. Now you'll note that in this case we have actin bound to ADP in an organic phosphate, and this is currently in what I referred to as, this would be my forward position. And you'll recall, just to quickly remind you, of the sketch that I drew in the other section. What I actually drew here is kind of the opposite direction. So I had my myosin, okay, and I had it drawn like this, and that was my forward position there. But in this case, when my myosin is going to cock and then contract, my actin will be pulled in that direction, as indicated by the arrow, whereas when we're actually going to contract here, my actin is being pulled in the other direction. So in essence, my figure that I drew for you is essentially a snapshot of what's going on right there, whereas the figure shown on the other side, on right here, is essentially going to be a snapshot of what's going on in this section here. The one of the other slides that I'll show you in a minute is essentially a upside down looking process. I'm going to show you what's rigor state using some pictures that I took from the internet, and that's essentially the upside down shown there. Now, so let's look at this. We've already talked about the role of calcium, but let's emphasize this and make sure everybody's on board. Again, I am being repetitive. If you don't want to watch this, skip it. That's fine. What I did on the whiteboard as I drew is sufficient. That'll get you through the exam, and that's just fine. So remember, muscle cells are released. Calcium is released from the sarcoplasmic reticulum. As calcium levels in the sarcoplasm rise, calcium then attaches to troponin. Troponin and calcium complex is formed. This causes a change in the shape. Anytime you see conformational, it means essentially a shape change. This change causes a change in tropomyosin. Tropomyosin glides out of the way, exposing the myosin binding site, and now myosin is allowed to access actin to form the cross bridges. All right, so we've already gone through that, so this is just a recap on the slides that you have available to you. Now we've talked about actin or myosin activation, and you'll recall that I talked about the steps of myosin activation. we have to activate myosin by attaching a molecule of ATP to it. So once actin is bound to ATP, it will then hydrolyze ATP into this inorganic phosphate and the ADP. So I'm coloring the ADP in a brighter purple or a brighter pink, and I'll color the inorganic phosphate in this blue color here. And so this process of activation is going to have the cell or the myosin move from the forward position to the cocked position. And you can see this is drawn a little bit differently than the way I drew it, but it's still the same method that is going to occur. And so this is essentially the myosin activation step that I spelled out to you on the other slides. And it's important to remember, this is something I will likely ask on the exam, is that this process here, activation, occurs when myosin binds ATP and then hydrolyzes that ATP to ADP, an inorganic phosphate, which causes it to move into the cocked position. Now then we have the sliding process. This is what's going to initiate the sliding. Cross bridge cycle, let's look at that. So here's my cocked position here with a cross bridge formed between these two portions here. And so this is my cocked position here. And then inorganic phosphate, let's look at that. Inorganic phosphate leaves, and you'll remember from the previous slide what happens when inorganic phosphate leaves. That causes a stronger attachment to the actin and then causes the power stroke. And so during the power stroke, we move from the cocked position into rigor state. So in this case, it's the forward position with a difference. In the forward position, myosin is not attached to actin, but in rigor state, it is attached to actin. So we still have this connection right here between these two protein molecules. Notice in this particular slide, ADP is shown as having as remaining bound, and this is consistent with what I told you. I'm not entirely sure whether ADP leaves during the power stroke or immediately after the power stroke. I have seen different textbooks say different things, and as I mentioned, I haven't really spent time digging into the scientific literature to find the answer. Maybe I'll do that one day, but I haven't yet, so I don't know yet whether it's during or after. And honestly, that part is not super important to me. I'm not going to get into the nitty-gritty on the exam. It might be important to somebody else, but to me, it's not that super big of a deal. What you need to know is that at some point, either during or after the power stroke, ADP leaves. And it has to leave because it competes with ATP for binding to that myosin. So if ADP does not leave, myosin cannot bind. Don't forget the term power stroke. This is an important term, and it occurs in response to inorganic phosphate leaving, exiting, detaching from myosin. And that detachment releases energy and causes myosin to move. Myosin is what we call a motor protein. Here's rigor state. It's upside down from what you're looking at. It's essentially the other side. And it's just because this is a good figure to show you rigor state. I didn't really have one in the textbook that I was using to make this in the past. Maybe I'll draw one day. But importantly, in rigor state, this attachment is firm and strong, and it will continue until this binding site right here. This is for the nucleotide binding site, and by nucleotide, it can either bind ADP or ATP. So ADP must leave in order for ATP to enter that site. Now, once ATP binds, it's going to cause the myosin to release. And as I mentioned, this rigor state is associated with rigor mortis. Here's the cross-bridge cycle as a whole, kind of looking at this in its entirety. And you can see here that we have each step. We start in the forward position here. We have this inorganic phosphate leaving, causing that bond here to become stronger, and also causing the conformation to change, so that we end up getting the power stroke. Please don't forget that it's leaving the phosphate, the phosphate leaving, exiting, detaching from myosin that allows the power stroke to occur. And then ADP, that's this pink little molecule right here. This is my ADP. It's released at some point, and now we're in rigor state. So this part right here is my rigor state. And then to break rigor state, we have to add my ATP. So they have this ATP being shown as this little blue ball right here. And so when ATP binds to that myosin, now we are ready for the activation process to occur again. And it starts by ATP, or by the myosin detaching from the tongue-tied there. The myosin detaching from actin, and then it hydrolyzes again, and we're right back into the starting the process all over again. And the cycle continues until calcium has been removed from the cytoplasm. The entire process of connecting calcium to the cross-bridge cycle is what we know as excitation-contraction coupling. I want you to be comfortable with this term, but that's what it means. It's essentially explaining this whole term, big long term, essentially encompasses this idea of calcium triggering contraction. And so, step-by-step process for excitation-contraction coupling is action potential in the neuron. I'm going to leave some steps out, obviously, for space requirement. Generates an action potential in the muscle fiber. Leads to calcium release. So calcium builds up in the sarcoplasm. And that is the process that initiates the cross-bridge cycle. So this whole collection of events are all part of what's called the excitation-contraction coupling. And we're going to refer to this process when I talk about summation, which is coming up. Summation occurs because of the time delay between calcium being released in the into the sarcoplasm and this cross-bridge cycle being formed. So there's actually a time delay between all of these processes, and that time delay allows for repeated excitation of the muscle, so repeated action potentials in the muscle to increase the amount of calcium in the muscle fiber, which will then result in a stronger and longer contraction. We'll look at that.